Well, I'll start by also thanking Hosian and the other organizers for this great opportunity to present my research to this group. Never has it been more apparent to me that I should be watching what I eat, exercising, etc. It's been driven home very well here. Although I'm going on the assumption of one of the posters outside that ethanol in moderation can actually extend your lifespan. So hopefully after dinner last night, that'll be true. So I'd like to discuss with whoever that is what moderation is. So today, the main message I wanted, we just heard a very beautiful talk on how mitochondrial ROS can induce age-related pathology. And what I want to show you today is that ROS are not always bad, that they can also be acting in a signaling mode that can be beneficial. So mitochondria have been talked about at great length here today, so I don't need to give a large introduction. But clearly, mitochondria are very dynamic and interesting organelles. They are comprised of many proteins, at least 1,200, if not upwards of 1,500 proteins. They are certainly at the crossroads of metabolism, having many different functions above and beyond oxidative phosphorylation. As we've just heard, they're a key component of the intrinsic apoptotic pathways. They're also involved in signaling and disease and aging, and it's really the nexus of aging and signaling that I'm going to be talking about today. As we also know, a critical component of mitochondrial function is the mitochondrial DNA. In humans, mitochondrial DNA encodes 13 proteins that are members of the oxidative phosphorylation complexes. It also encodes 2 ribosomal RNAs that are components of a complete different set of ribosomes that are in the mitochondrial matrix that translate the 13 mRNAs that are encoded by mitochondrial DNA. Also 22 tRNAs that are needed for translation. So it's a very important point here is that there's only 13 encoded here. In the oxphos complexes, there are approximately 80 proteins, so the rest of those are, of course, encoded in the nucleus and imported in. And I also want to point out that in terms of mitochondrial ribosome is really the only other constituent in cells, in mammalian cells and in yeast, that has components encoded both by the mitochondrial DNA and the nucleus. So mitochondrial ribosomes, the RNAs are encoded by the mitochondrial DNA, the proteins are encoded by the nucleus and imported in. So in these two cases, the electron transport chain, oxidative phosphorylation complexes, and ribosomes need to have coordination between the nuclear and mitochondrial genomes. Of course, the main function of the oxidative phosphorylation system is to make ATP. That's no surprise. We've also heard many talks showing that mitochondria also produce ROS. And in general, ROS are considered to be a damaging type molecule that, and its involvement in disease and aging is largely attributed to the ability of these to act as damaging molecules. But what I want to talk about today is the burgeoning field, really, of how mitochondrial ROS are used in signaling pathways. So in terms of signaling pathways, you can really define mitochondrial signaling pathways in 2 general ways. In the field, we generally talk about anterograde and retrograde signaling pathways, meaning, again, many of the genes are in mitochondria, most in fact are encoded by the nuclear genome. So changes in nuclear gene expression can transduce signals to mitochondria to change their composition and function. But when there's mitochondrial dysfunction or to maintain homeostasis of mitochondrial biology, you need to have signals that emanate from mitochondria and go back to the nucleus to lead to a homeostatic loops to maintain function or to change function. And these pathways have actually been pretty well delineated in yeast. There are transcription factors that are known to mediate the retrograde response, for example. However, in mammalian cells, retrograde signals really haven't received as much attention, and I point out that the types of things that can act in this way are calcium, ATP, for example, ROS are ones that I'm going to talk about today. In my lab, we've been focused for many years on nucleus-encoded factors that are required for expression and maintenance of mitochondrial DNA. And that includes dedicated RNA, polymerase, transcription factors, mitochondrial translation components, mtDNA replication and repair proteins, et cetera. But more recently, we've been really more interested in aspects of this nuclear mitochondrial signaling aspect. We have been studying basically 3 major areas in my laboratory, looking at the mitochondrial gene expression machinery. We've made links between those and deafness, which would have tied in nicely with Tomas's talk today, but I don't have time to talk about that today. We've also made connections between the ataxia-telangiectated mutated protein, which causes this disease, neurodegenerative disease and cancer, and mitochondrial dysfunctions involved in that we've demonstrated. But today I'm going to stick, and most of these studies actually began using yeast as a model system. Saccharomyces cerevisiae has a long-standing role in for identifying factors that are needed for mitochondrial function and regulation. And today I'm going to talk about some yeast studies in aging related to the TOR pathway. Okay, so the mTOR pathway, we've heard a little bit about. Suju Lin talked about it briefly. The TOR pathway is a very interesting pathway. First of all, it's a highly conserved pathway. It's conserved from yeast to man. It's involved in integrating both pro-growth signals and stress-resistance signals. So I thought this is really, and it's also a pathway, reductions in which lead to extension of lifespan in all organisms that have been tested so far. And I think the reason for that is twofold, in that what this pathway really does is it integrates signals from nutrients and pro-growth signals into protein synthesis and other important cellular pathways, but it also inhibits the stress response pathway. So it's really at the nexus of trying to decide, based on your nutrient status or your growth factor status, whether or not you go forward with a pro-growth pathway or you do stress resistance. And this, I think, is relevant to Bruce Ames' talk, where, you know, when you're limited for nutrients, you weigh on the side of going to your essential metabolic pathways and sort of forget about stress resistance. In reality, you really don't have enough energy in any particular time to do both. And I think that's the key to the aging nexus here. And I think the TOR pathway is really important in that regard. Okay, so I don't need to introduce this because Suju Lin talked about the models for yeast aging. And the model we're using is the chronological lifespan aging in yeast, where you look at how long a cells can live once they reach stationary phase. And the important point I want to make here is that in order to get there, we dilute the cells back from a culture and allow them to grow and reach stationary phase. And once we reach time zero here is the beginning of the aging curves that I'm going to show you. But the key point here is that there is this growth phase that happens before that, and that's where a lot of the action is that determines your lifespan later. That's really the main point of my talk. Okay, and we showed a few years ago that if you reduce TOR signaling in yeast, you extend chronological lifespan, and that's shown here. This is a typical lifespan curve of viability in stationary phase versus time, and you can see that deleting the TOR1 kinase leads to a robust extension of chronological lifespan. This was published a few years ago. We showed that this was absolutely dependent on mitochondrial respiration. So in row 0 strains that don't have any mitochondrial DNA and in other strains that are respiration deficient, TOR can no longer extend lifespan in those strains. This was also glucose dependent and oxygen dependent, which led us to believe that this was involved in nutrient sensing and perhaps reactive oxygen species. And in fact, if you look in stationary phase, the TOR1 strains have lower ROS as measured by DHE fluorescence by FACS. And they also have enhanced expression of some of the antioxidant enzymes here. SOD2, you can see, is upregulated at the protein level in the TOR1 null strains earlier than it is in a wild-type strain. So there's some connection between oxygen status, reactive oxygen species, and antioxidant defenses that's at the heart of what's going on here. So to try to get at the mechanism of this a little bit more, one of the main things that the TOR pathway controls is cytoplasmic translation. In fact, it's one of the most expensive things a cell does. So when you're low on energy, you turn off when TOR signaling gets lowered, and you reduce the amount of translation and ribosome biogenesis that's occurring in the cytoplasm to conserve energy. So one of the things we thought, well, simple-mindedly, is maybe TOR is controlling mitochondrial translation. And so what we did was you can specifically label mitochondrial translation products in mitochondrial DNA-encoded subunits by inhibiting cytoplasmic translation and feeding them methionine, labeled methionine. And you can see that during log phase growth, now before we start the lifespan experiment, that there is a robust increase in the rate of mitochondrial translation when TOR signaling is reduced. But that goes away, actually, in stationary phase. This leads to an increase in steady-state levels of both nuclear and mitochondrial encoded oxidative phosphorylation subunits. Here, subunits of cytochrome oxidase are shown. COX-1 is mitochondrial encoded. The others are nuclear encoded. But this increase in oxidative phosphorylation complex was not accompanied by an increase in the total amount of mitochondria in the cells. And so this was shown here by mitotracker green staining. We've done many Western blots to show this as well. There's no increase in the amount of mitochondrial DNA. And in stationary phase, we actually saw a decrease in mitochondrial membrane potential. To try to get at this a little bit more globally, we did a 2D gel proteomic analysis where we did 2D DIGE with highly purified mitochondria from both wild-type and TOR1 null cells. These were purified mitochondria, then you label with two different fluorescent dyes, mix them, and run a 2D gel. And by comparing fluorescence, you can find proteins in the mitochondria now that are differentially expressed, shown by these ones that are circled here. And the bottom line is this really confirmed for us that it was a global upregulation of the amount of OXPHOS complexes per mitochondrion that was occurring in TOR1 when you reduce TOR signaling. The other things we found that were upregulated are interesting. There's a highly abundant outer membrane protein of unknown function that was upregulated in mitochondria. There were several metabolic enzymes that we're following up at the moment. I can't tell you much more about that, but there's definitely some metabolic reprogramming happening. But one of the interesting things, especially with regard to Suju Lin's talk, is this protein YHB1 was upregulated, which is a nitric oxide detoxifying enzyme. So we're entertaining the idea that maybe not only ROS signaling is involved, but maybe nitric oxide signaling is involved as well. Okay, so we really became interested in this ROS phenotype, and it really became apparent to us that the increases in oxygen consumption that we were observing in TOR1 null strains and the increases in translation that we observed were occurring during this growth phase and not in the stationary phase. So that implicated events happening during that growth phase being determinant of what your lifespan is later in stationary phase if you're a yeast cell. And so we went back and looked at a lot of mitochondrial parameters during growth and stationary phase and compared them. And you can see here that mitochondrial membrane potential actually is higher in log phase growth and decreases as a function of the growth state. So it's higher to begin with and is lower when you go into stationary phase. So there's a dynamic regulation of membrane potential here. To get at whether or not these mitochondria, or what the functional capacity of these mitochondria are, we did some inhibitor studies. We used the ATP synthase inhibitor, TET, and you can see that in TOR1 null cells that the oxygen consumption could be inhibited by an ATP synthase inhibitor, indicating that they're coupled to a pretty significant degree, whereas the wild-type mitochondria really don't respond to that inhibitor at all. It's meaning that they're either not being utilized very much or they're not coupled. Similarly, when you add an uncoupler to these cells, the TOR1 null cells respond to an uncoupler, again suggesting they're more coupled than their wild-type counterparts. And in fact, we looked at purified mitochondria for respiratory parameters, and they are indeed more efficient and coupled. But I'm not going to show you that data. So this suggested that really what's going on is through this induction of the amount of OXPHOS complexes, we're getting a better coupled state in TOR1 null cells, and that's leading to what we predicted is an important determinant of lifespan. And so we looked at that more carefully. And so we asked to test the idea that it was actually coupling that was important. We did some lifespan studies where we added mitochondrial uncoupler just during the growth phase. So if we uncouple mitochondria just during the growth phase, you can see that that reduces the lifespan of a TOR1 null strain. So the more coupled state is actually important for the ability of TOR to extend lifespan. This had no effect on the lifespan of a wild-type strain. So it really suggests that the coupling is important for the extension of lifespan by reduced TOR signaling. If you add the uncoupler throughout the time course of the entire experiment, you get similar results, although you actually extend lifespan in a wild-type strain, which has been published by Barros et al. A few years ago. But you can really see a robust reduction in the lifespan because of uncoupling in the TOR1 null strain. So really, the TOR1 null strains really rely on coupling heavily as part of the lifespan extending phenotype. Finally, we looked at mitochondrial ROS, and here we're using DHE staining, again by FACS. And similar to the membrane potential, there are more ROS actually in early log phase growth and late log phase growth, but again, that goes down as you go into stationary phase. So this implies that increased ROS are our interpretation of this is that ROS are imparting an adaptive signal that's allowing a better stress response in stationary phase. Even though this is DHE staining, you can see that in the TOR1 null strain, the majority of that staining is in punctate structures in the cytoplasm, which co-localize with mitochondria pretty well, whereas in the wild-type strains, most of the staining's in the nucleus and in the cytoplasm distributed throughout. So it really looks like this is mitochondrial ROS that are being produced in the TOR1 null strain. So finally, we wanted to ask the question, is an ROS signal sufficient to extend lifespan? So we added metadione just during the growth phase. That's sufficient to extend lifespan in yeast. In a wild-type strain, actually has no effect at all in a TOR1 null strain because they're already making that signal. Finally, a prediction of this model to connect the ROS phenotype to the inhibition of TOR, we use the inhibitor rapamycin, which is a specific inhibitor of TORC1 signaling. And if you add rapamycin just during the growth phase, you can get an extension of median and sometimes maximal lifespan. And that leads to an induction of an ROS signal during growth as well. Finally, if you add rapamycin in stationary phase, it has no effect. So this really means that you need to have that the inhibition of TOR signaling, the important functional consequences of that with regard to lifespan are occurring during that growth phase and not in stationary phase. And there is an adaptive mitochondrial ROS signal of some kind that is being utilized to extend lifespan later. This was all published in Cell Metabolism a couple of weeks ago. And David Sabatini, this is David Sabatini's interpretation of our data, which I thought was pretty good. So he drew the mitochondria talking to each other. And if you, in logarithmic growth, see rapamycin to inhibit TOR signaling, they say, hey, maybe we should upregulate our stress responses because we might need them later. And later on, in fact, life is good. If they do not receive this signal, they're floating through growth phase, oblivious, if you will, to ROS signaling, and then later become unhappy and cause aging. So I thought that was a pretty good take on that. But if you could look at his commentary in Cell Metabolism, they actually didn't let him use this. He had to make them not talk to each other and make the mitochondria not have faces. So I thought that was a bummer. Okay, so what's our model? Our model here it's very well known. Walter Longo's group and others have shown that a major way that chronological lifespan is controlled in yeast is through stress response pathways and ROS detoxification pathways. TORC1, as I told you, regulates those pathways, and it regulates them through a PAS1 kinase called RIM15 and several stress responsive transcription factors, which are homologs of FOXO and somewhat related to NRF2. And these stress responsive factors get upregulated. They're normally inhibited by TOR, so when you reduce TOR signaling, these get upregulated and lead to a major portion of the ability to extend lifespan. But what we've shown that there's a separate branch of this pathway, that there is a direct effect on mitochondrial respiration and membrane potential that's generating an adaptive ROS signal during growth that we postulate is leading to a more enhanced stress response in stationary phase, and together these two give you maximal lifespan in yeast. And the implication here is that there must be an epigenetic or a very robust transcriptional response in response to this ROS signal that's driving what's going on, because events that are happening early are having an effect later. And so we've just begun to analyze this. One of the great things about yeast is that there's a ton of microarray analyses out there, and it just so happens to be 2 studies that are already published where they looked at the gene expression profile in response to rapamycin and metadione under the exact same conditions, and looking at gene expression changes early, soon there, 4 hours after those treatments. And we've taken those data across those lists and looked for transcription factor binding sites in the commonly expressed genes. And what we have found are some of the factors you might predict. This MSN2/4 complex gives one, but we've also found other transcription factors that are involved in growth on non-fermentable carbon sources, which induces mitochondrial biogenesis, oxidative stress responses. So we think these are candidate transcription factors that are mediating this adaptive response. And interestingly, 2 of them are Jumanji domain-containing proteins, which are known histone demethylases, which we think could be involved in epigenetic regulation of the genes, the stress response genes that are extending lifespan in this organism. So we're quickly following up that idea. All right. That's the end of my talk. I want to acknowledge three extraordinary graduate students who did this work: Nick Bonowitz, Elizabeth Schroeder, and Yong Pan. Thank you.
Thank you. Perfect timing, actually. 4 seconds shorter.
4 seconds shorter?
Yes.
I'll try better next time.
We have a question. So, Jerry,
Yeah, where are you?
What your data suggests is that if you transfer the yeast to anaerobic conditions in the stationary phase, it would even live longer. Is that true?
Yeah, so Peter Piper's group has actually shown, well, first of all, we showed that if you do it under hypoxia, you relieve the TOR effect. So there is an effect of hypoxia on that. Peter Piper group showed that if you actually go through a stage where you have upregulated respiration and then look at lifespan, that extends lifespan. So all of those sort of predict that preconditioning, if you will, is sufficient to extend your lifespan. And I think an anaerobic preconditioning would probably do the same thing. Enrique?
I don't know the meaning of this, but after the 2D gel and the analysis of different proteins, you have shown metabolic switch, and then there is a lactic heterogeneous in mitochondria. I mean, do you have an interpretation for the role of this LDH there? Might be related also to hypoxic conditions.
Yeah, it could be. We just haven't followed that up enough yet to be able to say convincingly what that is. One of those metabolic enzymes, interestingly, was in a different position on the 2D gel, suggesting there was a post-translational or a modification that was TOR-dependent that leads to its accumulation in mitochondria. So I think there's definitely a role for these metabolic switches and what's going on. Walter Longo's group has shown that in addition to the stress response aspect for extending lifespan, that the ability to metabolize ethanol is very important in the extension of lifespan. So my guess would be that those switches are involved in an enhanced ability to use ethanol, which is the main product of fermentation that they use after the glucose is gone. And so I think that's probably what's going on.
Yes, one question.
Putting together the two talks, Thomas presented previously that ROS production is increasing the aging process, is accelerating the aging process. And you show that the adaptation mitochondrial ROS signaling, which means that this is an important signal to extend lifespan.
Right.
So what?
So what?
So the ROS, reactive oxygen species, are good or bad? Because in one presentation it was, so the interpretation could be that in the Thomas model there are too much. So it's a question of amounts.
I think what's going on, I think the best explanation, I think Michael Ristow actually stated it pretty nicely, and that there is a certain amount of ROS when they're generated that are acting in a signaling capacity, and when those are enacted at opportune times, like during growth in my yeast case, or under caloric restriction when he did the worm case, that if you enact an ROS signal, that can adapt you to better handle high levels of ROS later. And I think that's what's going on. I think there are hormetic effects that are important for lifespan extension, and I think those can be enacted by an ROS signal at opportune times during development or during different aspects of cell growth. So I think in worms, actually, if you inhibit mitochondrial respiration just during the L4 stage of development, that's sufficient to extend their lifespan. So there really are developmental windows and opportunity windows where I think mitochondrial function in ROS is being monitored. And I think if you do it right, that'll lead to a heightened stress response later that can extend your lifespan by lowering ROS and other damages. That's what I think.
As you know, rapamycin added late in the lifespan of animal studies has shown a remarkable extension of life.
Yep.
So have you tested that kind of model in the yeast
Yeah, so you're referring to the mouse studies that were published last year by Richard Miller. And he showed that what they didn't do is do it early. So they did it relatively late and show that adding rapamycin can extend lifespan in many different backgrounds of mice. And I'd be really interested to know if you just added rapamycin early, if the extension wouldn't be more robust, which would be consistent with my yeast results. But when we added it in stationary phase, i.e. late in life, it had no effect. So I think there's a window there somewhere, and maybe he hit the tail end of it, and we're hitting the front end of it. And I think maybe if we messed around a little bit with the timing of rapamycin, that we might find an optimal window where that would work for mammals as well.